Let's look at the evolutionary developmental biology that underlines pigmentation. And in this particular case, let's look specifically at pigmentation of fly wings. So from this image, I think you can already tell that there's a lot of evolution of pigment patterns, both colors and where pigment is deposited in fruit fly and other fly wings. So this is a great model system to study how regulatory changes to genes and to the sequences that regulate where genes are expressed and when, those patterns and those processes can underlie this type of evolutionary developmental biology. Now, pigmentation in general is useful in a lot of ways to a lot of organisms, and that's one reason that this is a really powerful system to study, pigmentation in fly wings, for example. And that's because pigmentation in vast numbers of species can be used for all sorts of different things that are useful evolutionarily to improve fitness. For example, here are some. Pigmentation is used for camouflage to hide from predators. It's used in mimicry and in warning coloration. That is, some organisms evolve to look like more dangerous species to try to prevent predators from attacking them. Pigments are also not just used in the body for that sort of signaling, either hiding or signaling that you're poisonous or toxic or dangerous. Pigments are also used, for example, in our eyes. There are opsins, which are proteins encoded by genes that are used by our eyes to absorb specific wavelengths of light. And those help us distinguish different wavelengths of light and give humans, for example, trichromacy, the ability to detect 3 different wavelengths of light, and for our brain to integrate that information into essentially what we think of as a full color vision of the world. Although there are other species that also have ultraviolet sensitive opsins, so humans have red, green, and blue opsins, also called short wavelength, medium wavelength, and long wavelength opsins. So 3 different genes that encode 3 different proteins. Some species are tetrachromats. They have red, green, blue, and ultraviolet opsins. So some fish, some insects, and other species can actually see in the ultraviolet range, which humans can't. So those are also pigments that are proteins, in this case, that are produced by genes. They are chemicals, but they're not pigments the way we normally think of pigments like printer toner and printer ink, which are also pigments, but those are just chemicals that are produced by enzymes. For light perception, those opsins are proteins that themselves absorb wavelengths of light. Pigment that's produced by our enzymes in our skin cells, for example, offer us ultraviolet light protection. So there are melanic pigments, melanin, a dark pigment that absorbs ultraviolet light and protects the DNA in our cells from being damaged by that ultraviolet light, because the melanin, the pigment, blocks those wavelengths of light from getting farther down into our tissues. And there are lots of other reasons that pigmentation is really useful. Here, we might consider how pigmentation is useful in courtship. So today we're going to focus on the fruit fly species and genus Drosophila. So this is a phylogenetic tree of the, as it says, the Drosophila melanogaster species group. And this is from a paper by Artem Kopp and John True. And what they did here was they looked at the phylogenetic relationships between different groups of Drosophila. And again, these phylogenetic trees are drawn. This one's in a different orientation than you might be used to. It's also because I've rotated it for presenting in this slide. But the base of the tree is representing an ancestral state, or what the ancestor of all of these present day species, shown at the tips of the branches, look like. So an ancestor to all of these current species long ago is here, and then the relationships between the different lineages of flies are shown in these branching diagrams, where 2 branches that are very short and closely connected to each other indicate 2 individuals that are or 2 species in this case, that are very closely related to each other. So those would be sister taxa or sister species if these are species. So here's another group of most closely related individuals, and then this branch, this group of tips, is very distantly related to this group of more closely related species and taxa. The important takeaway from this tree is that there is pigmentation, what we're going to call a spot of pigment, in many different species. And there are some species where that male wing spot is missing. And those would be minuses if we looked at the figure like this. So it's only these species where we're seeing examples of the male wing that spot exists. And there are other species where that spot doesn't exist. The fact that some of these species and species groups like these two, and these two, and this one, which is pretty hard to see, but there is a little bit of dark pigment at the tip of the wing, and these ones over here, they're all very distantly related to each other. It would be easy to explain the evolution of this spot if all of those species that have it were the most closely related to each other. But this tree tells us they're not, because these two species that have a dark wing tip are very distantly related from that pair, and that pair itself isn't exactly most closely related to these species here. What this suggests, which we've seen before, is parallel evolution. The evolution of the same trait repeatedly, but independently. And so this provides an awesome opportunity to look at the genetic basis of the production of this wing spot. The ancestral state is inferred based on this phylogeny, that the most distantly related species and taxa did not have the wing spot, suggests that it evolved independently a few times throughout the evolution of the Drosophila genus. So the ancestral state is no spot. And so that suggests that this spot has evolved repeatedly. So as on the previous slide, this is another table from the same paper that shows different subgroups of species and the total number of described species in those taxa, the number of sampled species by the authors. So for example, in the Melanogaster group, there are 8 total species, 3 of which were sampled in this study. And they find that in the Melanogaster group, 0 of the species of Drosophila melanogaster have wing spots. So Melanogaster does not produce Drosophila melanogaster, the common fruit fly, which was often studied as a model system, doesn't produce wing spots. But other closely related species do. So for example, all 4 of the Drosophila suzukii species produce, or subgroup species, produce wing spots. And Drosophila biarmipes, which is going to be the focal species for our discussion, is one of those members of the Drosophila suzukii group. So it produces those dark wing spots near the tip of the wings in an anterior position. The males produce the wing spots, not females. This is a male-specific trait, just like the sex chromes that are produced on the front legs of these Drosophila. Later, these authors showed that the wing spot in males is an important pigmentation trait because it does affect courtship. It improves the ability of males that produce the wing spots to mate. And they did that with a simple mate choice test. So I'm showing the data from this paper here, one piece of data from the paper, where they measured the mating success as a percentage of attempted matings in males of Drosophila biarmipes that do have a wing patch with those that don't. So I find it actually kind of odd that they chose the black columns to represent the Drosophila flies that don't produce the black spot on the wing, so don't get confused there. But the important point is, within the first hour, the males that did have a wing patch were twice as successful in general in mating as the Drosophila males that did not have this wing spot. But that was only a short-lived effect because over time, all of the males were essentially able to 90% of the time or so mate. So this is an evolutionary important change in pigmentation. The spot matters. The wing spot, the pigment spot in Drosophila biarmipes does have a fitness effect. It's useful when it's there, and it's not useful when it's not. So that's why this is a really useful evo-devo system to look at. How did this patch evolve? So we need to know a little bit of something about the genes and the proteins and the pathways that allow pigmentation to be produced in Drosophila. And in this case, we're going to be focusing on the yellow mutant, which is a mutant fly that doesn't produce the normal amount of black pigment or melanin that are present in wild type Drosophila flies. And thanks to a paper by many, but including this one by Trish Whitcop, John True, and Sean Carroll, we know a bit about the pathway that is important for producing pigments in flies. And this is the pathway that starts with the amino acid tyrosine, and that's converted. So this is not a gene pathway. This is a biochemical or an enzyme pathway. So these are not genes now. Tyrosine is a chemical that's processed by an enzyme TH, I believe tyrosine hydroxylase, into DOPA. And that DOPA can go down one of two pathways. It can be processed by the yellow enzyme, which we're also going to denote just with a single letter Y, into dopamelanin, which is melanin. It's a black pigment. DOPA can also be processed by DDC, dopa decarboxylase, I think, into dopamine. And the authors are indicating that we don't know what pathway produces a brown pigment, dopamine melanin, from dopamine, but they do know that ebony, another enzyme encoded by a gene called ebony, and we can denote ebony with a lowercase e, produces NBAD from dopamine. And NBAD is then producing a yellow pigment, NBAD sclerotin. Wow, NBAD sclerotin. Probably pronouncing that wrong. First time I've ever heard of that. So, the important point on this slide is to understand that something is known about how yellow pigment is produced and how black pigment is produced. And here again, we see this really confusing nomenclature where the protein yellow, encoded by the gene yellow, produces black pigment, and the protein ebony produces yellow pigment, and that's, again, because these genes are named after the mutant phenotype. So if you are a fruit fly and you don't have the yellow gene, and you don't make the yellow protein that can produce black pigment, what happens instead? The DOPA becomes, through this second parallel pathway, this is one way that DOPA can be processed. The other way is to the right. So it becomes dopamine, and then it gets turned into NBAD, which is a yellow pigment. So loss of the yellow gene creates fruit flies that don't have any black pigment. And likewise, if the ebony gene is lost or mutated or removed, then the flies can't produce yellow pigment, so they become black or ebony. So we're going to see these genes a lot more. But here are the phenotypes that the authors described from this work. So wild type Drosophila melanogaster. Now this is melanogaster, this is not biarmipes, the species that produces the wing spot. Drosophila don't produce wing spot. Their wild type phenotype is shown here in part of the body of the fly. But then we're going to focus on looking at the wings. You might be able to tell that there is some black pigment. It looks kind of grayish, but there is black pigment that's produced, not heavy amounts of black pigment, in the wild type Drosophila melanogaster wing. And that's more evident when you look at the yellow mutant. Remember, the yellow mutant is mutant in the yellow gene, which normally produces melanin, the black pigment. So when the yellow protein is absent, then that dark pigment that's normally produced isn't made, and so the wing looks a little bit yellower. It's not as dark. Likewise, when the ebony gene is mutated, the wing becomes darker. There's more pigment produced. There's less of the dopa that turns into a yellowish pigment. It's all shunted into the ebony producing or the melanin producing pathway. So those are some examples of how these proteins work to process dopa and produce pigments in the wings and the body of the fly. And ultimately, what this suggests is that it might be the yellow gene itself that's responsible for an increased concentration of black pigment in the wing of Drosophila biarmipes. So we're going to have to see if that is indeed the case. One technique that the authors are going to use to identify where in the body of the animal a protein is located, like the yellow protein or any other protein, is immunohistochemistry, sometimes abbreviated IHC. And the way that this process works is typically a tissue, a thin slice of tissue, or perhaps an entire wing of an organism is adhered to a glass slide. So these are often tissue sections, a thin slice of tissue. And then antibodies are applied to that tissue. Antibodies are often indicated with this sort of a structure, where this is the antigen recognition site. That is the antibody itself is a protein. It's produced by genes in our bodies. And the antibody has essentially two parts, this constant domain at the top, and then the antigen recognition portion of the antibody. So this is amino acid. This is a protein structure that specifically recognizes and will bind to a specific shape of another protein, the antigen. And in particular, that's referred to as an epitope. An epitope is a part of a surface of a protein that an antibody will recognize. So we could imagine, for example, that there's a protein in blue that has a particular structure, and in this case, this antibody is specific to that structure. So it will bind wherever this protein is found. And that's exactly what's happening in all of these figures, where an antibody, a primary antibody, which just means it's the first antibody, primary, to be applied, is added to the slide. So that's what these little red blobs indicate, is that this primary antibody has been added to the slide, and it will stick to the tissue, essentially, wherever its antigen, the protein, is found, wherever its epitope is found. So then the next question is, how do the scientists visually see where their antibodies have gone? Because the point of this is to visualize where in this tissue a specific target protein is located. So there are a few different ways to do this. In primary labeling, that antibody is conjugated or covalently attached to some sort of a molecule. Maybe it's one that produces light. So that's one way to visually see where the antibodies are located after they've been applied to the tissue section. Another approach, which is more useful because there are so many different antibodies that can be produced to so many different epitopes on so many different proteins that are produced in every species that a scientist might want to study. So there are lots and lots and lots and lots and lots and lots of primary antibodies. But it's difficult to covalently modify all of those individual antibodies that individual scientists are having produced for their favorite proteins. So it's difficult to make primary antibodies that are directly labeled. There are very few of them. So what's more useful, because it allows for combinatorial work, is to produce the primary protein, the primary antibody that recognizes the specific epitope protein that you're interested in. And then, because the constant region of the antibody is specific to the species that it's made in, so let's say this was a primary antibody that was produced in mouse. That means that the constant region of this antibody has a particular protein shape that another antibody could recognize. So then the authors will apply, in this example, a second antibody, this is a secondary antibody, whose antigen recognition surface is designed to recognize mouse primary antibodies. And so that secondary antibody is the one that's conjugated to, for example, a light-producing molecule or a fluorescent molecule that's easy to observe microscopically. So that's indirect labeling. The first antibody recognizes the antigen. The second antibody is the one that's coupled to the chemical or enzyme or something that allows the scientist to observe where that complex is located on the tissue culture slide. And the reason to do this again is because it's easy to make secondary antibodies that recognize any mouse antibody. And so companies can manufacture these and couple them to all sorts of different fluorescent molecules or enzymes. So this is, for example, let's say the secondary enzyme was produced in goats. This would be called a goat anti-mouse antibody. It's a goat antibody that's produced in goats, and it specifically recognizes mouse antibodies. So that's indirect labeling. And then they're also indicating this last option, which is indirect labeling with signal amplification. So in that case, the primary is applied, the secondary is applied, and the secondary is connected to a huge molecule with lots of different surfaces that can be bound. So in this case, this example is indicating this is a biotinylated secondary antibody. And what that means is that then there is another protein, avidin, which recognizes and binds to a lot of avidins bind to a single biotin molecule. So that's the reason that this is called amplification, is because each of those avidin molecules is itself coupled to some sort of light-producing or chemical-producing enzyme molecule. So you get a huge amount of light or pigment production or whatever sort of signal it is that's coupled to the avidin. So this is basically a way to make the fluorescent signals or light signals much brighter in immunohistochemistry, so they're easier for scientists to detect. And in this example, from again the Whitcop, True, and Carroll paper, here, this is another figure from their paper. They were interested in understanding where in the fly is the yellow protein produced and where in the fly is the ebony protein produced. So they're using here immunohistochemistry to identify where yellow is located, and they see an increased concentration of yellow right there where the wing spot is produced in Drosophila biarmipes. And interestingly, they also find when they use anti-ebony primary antibodies, they see an absence of ebony protein where they see the presence of extra yellow protein. And again, this makes sense because if there's a black spot, you would expect the yellow protein to be wherever that black spot is located, producing melanin. And we would expect not to see ebony produced there, because if ebony were in that wing spot, it would be doing this opposite or antagonistic approach of removing some of that DOPA from the melanin production pathway to produce yellow pigment. So again, the big question is, how did Drosophila biarmipes get its wing spots? What's happening at the gene regulation and protein production level of, say, the yellow enzyme to concentrate, apparently, yellow in these spots? And there are there are at least two, maybe more, models of what might be happening in the wing of the Drosophila to produce this particular spot. So one possibility is that yellow, which again we're calling lowercase y, is produced everywhere in the wing of the fruit fly. But perhaps there's also a yellow repressor that's also produced everywhere in the wing. So yellow protein is trying to be turned on, but there's a repressor protein that's turning it off. Except for, perhaps, what if there's a Y repressor? I know this is going to get complicated. So the Y repressor would repress the yellow repressor, which itself presumably represses Y. So what if there's this protein that's a repressor of yellow, and it is only expressed in that spot. So that's the only place in the wing where yellow isn't being repressed. And so that's the only place where we see yellow protein activity and a pigment being produced. So that's one complicated but possible reason for the production of a small region of pigment in the wing. The other possibility is a bit more straightforward, which just says that there's some sort of regulation in the wing tissue that makes the yellow protein only ever expressed in that part of the wing, not in any other part of the wing. So those are two different possible models for how developmentally this sort of a pattern could be produced. So in conclusion, how are scientists likely to be able to figure out what enhancers or transcription factors, promoter sequences, are important in regulating the production of this specific pattern, this wing spot? Well, assuming that yellow is involved, what the authors of any study, what scientists might want to do, is to look for transcription factors that would presumably bind to a promoter or an enhancer, either. More likely an enhancer than a promoter. Remember, enhancers and promoters are both DNA sequences. Promoters are near the transcription start site, and enhancers can be anywhere, upstream, downstream, another chromosome perhaps even. So the transcription factors that regulate yellow expression themselves might be predicted to be localized to the wing spot. So one approach would be to look for transcription factors that have the same protein expression pattern as the genes and proteins they regulate. So if yellow, for example, is only produced in this spot, we would expect to find maybe a transcription factor that itself is only expressed in that spot. So that would be taking this process one step upstream, essentially, or one step prior, that the transcription factor is only produced in that spot, and therefore yellow is only produced in that spot. So that would be more like the second model that I just described, where if yellow is only produced in the spot, then the authors or scientists would look for probably a transcription factor with a known binding site in an enhancer or the promoter of the yellow gene. The other thing to do would be to look in a different species. So that could be Drosophila biarmipes. What if we looked at the same region of Drosophila melanogaster, a species that doesn't have wing spots? So it still has the yellow gene, which produces the yellow protein, which produces black pigment in Drosophila melanogaster. But we might expect it not to have a DNA sequence that allows that same transcription factor, whatever it is, to bind to the Drosophila melanogaster enhancer, or at least not in that region. So we might also be able to, say, clone that regulatory sequence from Drosophila melanogaster and put it in Drosophila biarmipes and see if that gets rid of the wing spot. But that's a harder experiment to do because biarmipes already produces a wing spot. So then you'd have to be trying to design an experiment that gets rid of the wing spot, which is not usually easily done by transferring DNA from one species to another. That would involve more like mutagenesis in biarmipes to try to destroy the DNA sequence that that transcription factor presumably, putatively binds to turn on yellow production and the wing spot. So another type of experiment that could be done would be to take whatever enhancer or promoter sequence from biarmipes, the species that does produce the wing spot, and to put that into Drosophila melanogaster, a species that doesn't normally produce a wing spot, and see if Drosophila melanogaster then produces a wing spot. So by cutting up little pieces of the enhancer and promoter regions of the yellow gene in biarmipes and moving that into Drosophila melanogaster, you might be able to test something. And I'd like you to think, is that a necessity test or a sufficiency test? You might be able to discover if there's a region of the regulatory sequence around a yellow gene that can actually turn on the production of a wing spot in the same way in a species that doesn't normally produce a wing spot. Now, the last thing I want to mention is about reporter genes. And in this case, scientists often use green fluorescent protein, or GFP, which is a gene that produces a protein that fluoresces a green color. So GFP makes messenger RNA for GFP. And when that's translated, it produces a protein that has a particular shape that it can absorb certain wavelengths of light and emit green light that's easy to see. So in the case of yellow, if the authors find a putative enhancer, putative means potential, that regulates the specific wing spot pattern, they can put that enhancer upstream of GFP reporter gene. So this again is an example of a reporter gene. It's not a gene that's natively found in the species it's being studied in. It's just being used as a positive indicator of where this enhancer will act. So in other words, what you can do is you can insert this construct, which I'll circle in blue. So this DNA construct would probably be a plasmid, or it might be integrated into the genome of the Drosophila melanogaster. So you put that in a Drosophila melanogaster wing, right? These flies don't normally produce wing spots. But if this enhancer region, say that's from Drosophila biarmipes, BIA, if that enhancer is important in that wing spot production of yellow, if you put this construct into Drosophila melanogaster, maybe when you find an enhancer sequence that is critical for producing the expression of whatever gene is being regulated by it, normally in biarmipes that would be yellow, but here we're using a reporter gene, GFP, the authors and scientists might hope to see that they see, that's yellow, not green, but I'm colorblind. They might see GFP expression. Here, let's try green. In Drosophila melanogaster of this reporter gene, only if the enhancer that they're using to regulate expression of the reporter gene is relevant to and is causing that localized expression of whatever that gene is, be it yellow natively in Drosophila biarmipes, or be it GFP, a reporter gene in this artificial construct that's put in Drosophila melanogaster. So that's a little bit about immunohistochemistry and about the use of reporter genes to detect both the location of expression of proteins and also the regulatory sequences that drive those expression patterns.